Good morning. A very good morning, one and all. Welcome back to the second day of International E-Conference on Emerging Trends in Bioscience and Technology 2020. Very soon we are starting with our today's session. Today we have very distinguished guest with us to glorify this today's session. To introduce our guest speaker for the second day, I would like to invite Samiksha Kandil from Biotechnology Society of Nepal. She is a very young and energetic organizing secretary of this Biotechnology Society of Nepal. Now I will request Samiksha Kandil to introduce our today's guest. Over to you, Samiksha.
Thank you so much, ma'am. A very warm good morning and namaste to all of you. So I'm going to start the session. I hope we are audible to you all. So our first guest speaker is Dr. Gopal G. Gopal, Assistant Professor of C.G. Bhakta Institute of Technology, Uttar Tarsedia University, Gujarat, India. Let me introduce him to you all. A graduate of biotechnology from Gujarat University, who holds PhD in from Center for Molecular Medicine in the structural feature of type 4 secretion system. He is also an author of a lot of journal article publications, and two books has already been published. He holds a number of recognized awards from India, including Fast Track Young Scientist Award, Dr. D.S. Kothari Postdoctoral Fellowship from UGC, several awards from CSIR Board, National Merit Scholarship for pursuing MSc in Biotechnology from Department of Biotechnology, and a full fellowship in BSc from CM College, India. Apart from this, he has presented a number of research as posters, as well as delivered a number of invited guest lectures as a resource person in more than 65 events. Along with this, his training experience has also been nationally recognized and is involved as advisor in many private limited around India. Along with this, he is also got many experiences of coordinator as well as convener in many national and international seminar, conferences, and workshop. So let us welcome him to our session. So to deliver his glorified lecture entitled Role of Biotechnology in the Management of Diseases. Hello, sir. Good morning.
Good morning. Am I audible to all?
Yes, sir. You are very soundly audible. Please start your session.
Good. So good morning, everybody present online. And I would like to thank organizer, especially Basdev Jamare sir and his team for utilizing this lockdown period for dissemination of knowledge. So I'm sharing my PPT, just wait. Hello. Are you able to see? Hello. Hello. Are you able to see me?
Yes, sir. Yes, sir. Your PPT is visible and you are audible too. You can continue.
Okay. So just keep on responding sometime.
Yes, sir. Please.
Sometimes internet will go or light will go, so I should know whether you are listening or you are watching or not.
Yes, sir. We will communicate you immediately.
Good.
Thank you.
So today's my topic of talk is role of biotechnology in the management of disease. As madam have already introduced me, so no need to introduction again. So what is biotechnology? So using living organism or material for commercial, industrial or healthcare purpose. So we use that biotechnology defined differently by different organization, but whenever we use living organism for our benefit, we call it biotechnology. So biotechnology is the use of biological process, organism, system to manufacture products intended to improve the quality of human life. Today we will focus on one major part of biotechnology, that is gene manipulation technology, or we can call recombinant DNA technology, gene cloning technology, because we all know biotechnology because of this gene manipulation technology. Gene manipulation technology revolutionized the biotechnology and other field of bioscience also. So as a brief introduction, a gene is a basic functional unit of heredity, and gene are segments of DNA and having information in the form of nucleotide sequence, which is decoded when required in the form of mRNA and proteins. So gene directly or indirectly is responsible for every character of an organism. So whatever character you have or any organism have, that is directly or indirectly controlled by gene. So if we wish to add some character in organism which doesn't have, so we can add that known gene to that organism and then we can get that character. So basis of gene manipulation technology is this universality of this pathway. If this pathway is not universal, then gene manipulation technology could not be a successful technology. As you know, codons are universal. Means if this gene is coding a particular protein in microorganism, then it will produce or it will code same protein in plant and animal in other organism. That is universality. So codon universality and this decoding pathway, that means the gene decoded to that mRNA and then to protein. So this pathway is almost universal in all organisms across the kingdom, and the codons are also universal. That's why gene manipulation technology become a successful because of this universality. So what is gene manipulation? In briefly, we can say that we can isolate desired gene from an organism, and then there is vector which can carry your desired gene. So we isolate usually from bacterial source, and then we ligate both desired gene as well as that carrier vector, and then again we transfer that ligated product recombinant DNA into the host so that it can replicate, it can produce high copy of that gene. And there are basically two main purposes of this gene cloning. Either we need more amount of that gene cloned, or we need proteins coded by that gene which we have cloned. So basically two purposes of gene manipulation technology or molecular cloning. Now, as per my topic of talk today, how disease can be managed, how disease can be reduced. So if we provide nutritional food to all citizens, all human, then there is less chances of disease. So nutritional food can overcome the disease caused by malnutrition, like mineral deficiency, like protein deficiency, like vitamins deficiency. These things cause many diseases. So if we provide nutritional food, mineral containing food, vitamin containing food, then we can overcome, we can reduce the burden of disease. Second thing, prevention of pathogenic disease. So if we prevent pathogenic disease either caused by bacteria or virus or some other organism, so by vaccination we can reduce burden of that disease. As you know, there are several vaccines available. We will discuss in the next slide. Diagnosis of disease. So if we diagnose disease accurately, then we can handle that disease, we can treat that disease effectively. So diagnosis of disease, accurate diagnosis of disease is very important. Treatment of disease, if we diagnose the disease, then we must have that treatment methodology, we must have drug to treat that disease. And fifth, understanding the new or unknown disease. So if there is occurrence of new diseases either due to pathogen or due to gene abnormalities or for other reason, so we must understand new diseases. So in this way, in 5 ways, we can manage the disease, or we can manage the burden of that disease. We can reduce the disease burden in that citizen or that mankind. So how gene manipulation technology, biotechnology is playing a role in all these 5 strategies to reduce the gene means disease burden. So let's see. So to produce nutritional food, there are several examples available, and scientists are trying to produce more such type of foods. Like golden rice, this is very good example. A transgenic rice having high vitamin A content, so you don't have to take extra pill of vitamin A if you will take that golden rice. So through transgenic methodology, gene manipulation technology, scientists have added carotene biosynthetic gene in that golden rice, that paddy plant. So in addition to that carbohydrate and other things, you will also get sufficient amount of vitamin A if you will consume that golden rice. High iron containing transgenic banana. So now banana is available which have high iron content. Natural banana have also high content, but scientists have added ferritin gene so that banana can store iron, and if you will consume that banana, you will not feel deficiency of that iron. Transgenic soya with high amount of vitamin D. So scientists are producing different nutritional food items by transgenic methodology, by gene manipulation technology, molecular cloning. So we are getting nutritious food through recombinant DNA technology. Second, how can we prevent disease? By applying gene manipulation technology. So production of different categories of vaccines. As you know, so there are different categories of vaccines. Either we can use attenuated pathogen, or we can use subunit vaccine, or we can use DNA vaccines. So for all these types of production of all these types of vaccine, we have to apply gene manipulation technology. And there are several vaccines available which prevent occurrence of that disease, pathogenic disease. Vaccines are the administration of antigenic material to stimulate an individual's immune system to develop immunity against pathogen. So since you all are bioscience teacher or participants, so as you know, vaccines are nothing, it's dummy pathogen, so that our immune system become elicited. It becomes activated. So when original pathogen will infect, our immune system will fight with that pathogen effectively, efficiently. So this is antigenic, whatever antigenic things we administer in our body, so that our body become immune to that proteins or that bacteria or that pathogen. So as we discussed type of vaccines, we can use attenuated vaccines, inactivated vaccines, subunit vaccines, DNA vaccine. For subunit vaccine and DNA vaccine, we completely use gene manipulation, molecular techniques, molecular cloning techniques. So we can produce vaccines using that biotechnology or recombinant DNA technology. You can see scientists are using virus code protein, like in COVID, that in coronavirus, still scientists are trying to produce vaccine against S code protein of that coronavirus. So we again use that, if we know their gene sequence of that spike protein, then we can produce vaccine against that code protein. For production of vaccine, we have to apply recombinant DNA technology methods. You can see impact of vaccines. In 1950s, there were 5,796 known cases of diphtheria. But after vaccination, you can see in 2009, there is 0 cases of diphtheria. Like tetanus, 486 were known cases in 1950. But in 2009, there is only 18. And pertussis, 1,20,000 known cases in 1950. Now population have increased 3 times, but still you can see there is huge reduction in that cases of that pertussis, measles, mumps, rubella, hepatitis, hepatitis B, and there are several other vaccines. So you can see the impact of vaccination. If we vaccinate, then there is very huge reduction in the occurrence of that pathogenic disease. For in diagnosis, how can we use recombinant DNA technology or biotechnology for diagnosis of disease? So as you know, PCR is universal methods. So there are several PCR based diagnosis techniques are applied nowadays. PCR, RT-PCR means if some viruses are having RNA, then we use RT-PCR. If we wish to find out load of pathogen, load of that gene, then we perform real-time PCR. So these PCR, RT-PCR, real-time PCR, and there are different multiplex PCR, state PCR, and there are several PCR techniques we scientists using to diagnose the presence of pathogens, either bacteria or virus, or sometimes to find out abnormality in genome or gene. So PCR is the very, very efficient technique to diagnose the disease. Antibody-based diagnosis of disease, there is immunocytochemistry, immunohistochemistry. So you can see wherever there is immuno is added as a prefix, you must know that antibodies are used to find out presence of some proteins. So we can find out presence of pathogen, bacteria or virus or some abnormal protein by using antibody-based diagnosis. And how can we produce antibody? So we can produce antibody by producing recombinant protein, because recombinant protein we can produce in high amount, in good quality, purified protein, and we use that recombinant protein to raise antibody. Whether polyclonal antibody or monoclonal antibody, we must have purified protein, high amount of protein. So most of the case, we use recombinant protein to produce antibodies. And as you can see, antibody, there are several techniques which are antibody based. So DNA hybridization based diagnosis, southern blotting, northern blotting, FISH, DNA microarray, these techniques are based on DNA hybridization. So we must have that DNA. We must have the probe, then we can perform these techniques, DNA hybridization based technique. DNA sequencing based diagnosis, single nucleotide polymorphism, you must have aware of about that SNP. SNP is causing disease in several patients due to abnormality in sequence. There is abnormal protein production, so there is cause of disease. So we find out single nucleotide polymorphism. We can use gene sequencing. We can find triplet codon repeats, like in Huntington disease, there is CAG repeats. So we can find out that abnormalities by DNA sequencing. There are different alternate methods also. So for diagnosis of disease, which is required for efficient treatment of disease, we use molecular biology or recombinant DNA technology. As you know that PCR is very versatile because of two unique features of PCR. In PCR, we increase quantity of given DNA to million or billion copy. So that wherever there is less quantity, we cannot observe it, we cannot analyze it, then we do PCR so that it will increase to million or billion fold. So we can easily handle that DNA, we can easily analyze that DNA. Second thing is, in PCR, we amplify only a specific sequence from mixtures of DNA. So there is a specificity. From mixtures of bacterial genome, we can find out presence of one gene from that bacteria. So we can efficiently find out pathogen, presence of pathogen, presence of some abnormal gene using that PCR. You can see immunofluorescence. So to find out presence of some protein at cell surface or tissue surface, we can do that immunofluorescence technology, but we must have antibody against that protein. Then only we can perform this type of techniques, either Western blotting or immunofluorescence techniques. Now, so in treatment of disease, how can we use recombinant DNA technology in treatment of disease? So as you know, we can produce therapeutic recombinant protein. Several human disorders are due to absence of or malfunction of proteins usually synthesized in the body. Human proteins are produced in bacteria or other organisms using recombinant DNA technology. There are a huge number, hundreds of recombinant protein we use as a therapeutics to treat the disease. Those proteins generally we produce in bacteria or sometimes in yeast or sometimes in mammalian host cell. So if there is some disease due to absence of some protein, we can produce that as a recombinant protein. So for these things, again, we have to apply recombinant DNA technology, gene manipulation technology. Therapeutic nucleic acid used for gene therapy. So addition or deletion of gene which cause disease or mask the expression of disease causing gene. So we use antisense technology, gene therapy technology if we have therapeutic nucleic acid. So we produce therapeutic nucleic acid, DNA or RNA using again recombinant DNA technology. Therapeutic antibodies, as you know, for cancer treatment, for other treatments, we use monoclonal antibody. So one of the example are antibodies conjugated with toxins, which we use to kill cancer cells. Antibiotics, as you know, there is huge role of antibiotics in treatment of pathogenic disease, especially bacterial disease. So antibiotics are produced by microorganisms. But to increase yield or get modified version of antibiotics, scientists apply gene manipulation technology. So generally it's naturally produced by bacteria, but through gene manipulation technology, we increase yield of antibiotics. We use biosynthesis, rate of biosynthesis of that antibiotics. We can also increase biosynthesis and production of natural drugs like vinblastine and vinblastine. This is natural product, but through metabolic engineering, we can increase its production. We can increase its yield per unit mass of that plant or that bacteria through gene manipulation technology. That is generally we call it metabolic engineering. So you can see there are a list of therapeutic proteins which are in commercial use. You can see there are different ways of gene therapy. If there is weird and tiered gene is present, then we can replace it. We can increase number of copies. We can also kill cell by adding some drug susceptible proteins at the cell surface. So there are different ways of gene therapy we can use. For all type of gene therapy, we must have that gene. We must have high copy of that template DNA gene. These are methods of gene therapy, generally ex vivo. We also use in vivo by viral mediated transfection. So there are list of therapeutic monoclonal antibodies. As you can see, Abseq, CMAB, adalimumab. MAB means monoclonal antibody. That is suffix generally scientists use. These are all trade name of monoclonal antibodies. And there you can see target disease, percutaneous coronary, rheumatoid arthritis. These diseases are treated by monoclonal antibody. So if due to abnormality in protein, high amount of protein production, then we can use different monoclonal antibody to suppress the effect of that protein. So we use monoclonal antibody, we produce monoclonal antibody again by using recombinant DNA technology. These are methods of production of monoclonal antibody. Since we have only 30 minutes, so I'm going through the things so that in future you can apply these techniques, and you should also understand the potential of recombinant DNA technology. Recombinant monoclonal antibodies. So generally we produce monoclonal antibody by hybridoma technology, but nowadays scientists are trying to produce monoclonal antibody using recombinant DNA technology. Like if we know the gene coded by that monoclonal antibody, then we can produce that monoclonal antibody in bacteria also. So rate of production of monoclonal antibody will drastically reduce. So scientists are trying to produce monoclonal antibody in bacteria. Generally, we use hybridoma technology. We use mammalian cell to produce monoclonal antibody. But recombinant DNA technology or recombinant monoclonal antibody also we can produce. As you can know, the impact of discovery of antibiotics, it is estimated that penicillin has saved at least 200 million lives since its first use as a medicine in 1942. So only single ampicillin, penicillin saved 200 million, means 20 crore people. Nowadays, there are different variants of antibiotics are available, and we increase yield also through metabolic engineering. As you can see, how can we increase drug biosynthesis or antibiotics by metabolic engineering? So either we can remove rate limiting transcriptional or allostatic control, kinetic enhancement of rate limiting enzymes, genetic inhibition of competing pathways, enhanced carbon commitment to the primary metabolic pathway from central metabolism, modification of secondary metabolic pathway to enhance energy metabolism and the availability of enzymatic factors, enhanced transport of the compound out of the cell. In this way, we can increase normal biosynthesis of that drug or some small molecule or antibiotics from natural hosts. We don't have to isolate that gene and add into other host, because sometimes suppose antibiotics produced by several enzymes. This is not one enzyme product. In that case, we add some genes so that if there is some rate limiting steps, so we add that extra enzymes, extra gene, or we sometimes remove some bottleneck effect by adding or removing some genes. We can increase their natural biosynthesis. So in this way, we can get more yield of that antibiotics or that drug from that natural plant or natural microbes. Understand the cause and mechanism of disease. So if you don't know if you don't know new disease, what is the reason, whether pathogenic or some genetic disease, in that case also we use gene manipulation technology to understand the disease. Like we produce transgenic animal as a model of human disease. So we create disease mice, rabbit by deletion or addition of gene to understand the disease. We have some examples: creation of model of Alzheimer's disease by overexpression of amyloid precursor proteins, creation of mouse model by knockout for hypoxanthine guanine phosphoribosyl transferase deficiency to study Lesch-Nyhan syndrome, which is neurobehavioral abnormalities. So in this way, we can produce model organism by deleting some gene or adding some gene, so we can understand the disease, or we can understand the efficacy of some drugs on that model organism. So we must have model organism to understand the human disease. To produce that, again, we have to use gene manipulation technology. Gene targeting has been widely used to model human cancer caused by the inactivation of tumor suppressor genes such as TP53 and RB1, finding the function or role of unknown gene. So this is very important. As we all know, since there are next generation sequencing is available, scientists are dumping the data of genome sequence, but they are unable to find out role of that gene. Whoever have that genome sequencer, they produce that data, but they don't know what is the role of gene. To find out the role of gene, again, we have to apply recombinant DNA technology techniques like gene knockout. How can we find out the role of gene? So by knocking out the gene or knocking down the gene or producing the protein coded by that unknown gene, and then we characterize that protein either by crystallization or enzyme assay, whatever way. So we must have that protein. So we use recombinant DNA technology to find out role of unknown gene if gene sequence is known. Pharmacogenomics, so role of genome in drug response. So if we add some drug, so we can find out what is the repercussions, what is the abnormalities, or what is the effect of that drug on gene, on genome. So for that also, we must have that gene manipulation technology. We apply that gene manipulation technology to find out the role of drug on gene. So in this way, to understand the new disease, mechanism of disease, we apply gene manipulation technology or recombinant DNA technology. So as a summary, you can see we can provide nutritional food for prevention of disease. We can produce immune vaccines. We can produce, nowadays you are hearing about that serum therapy of COVID-19. So in serum, there is antibodies. So scientists are trying to use that antibody. So that is passive immunization. Diagnosis, PCR-based, blotting-based, ELISA-based, Western blot-based. So for diagnosis also, we use recombinant DNA technology. For treatment, we can produce therapeutic proteins, oligonucleotide, antibodies, antibiotics through recombinant DNA technology. To understand the disease, creation of disease, animal model, pharmacogenomics, all these diseases. So I think I have finished within time given allotted to me within 30 minutes. If you have question, so I think there are lots of questions, then I'm happy to reply all those questions within my time. Thank you. Hello. Hello.
Hello,
Have you?
sir. Are you
Yes.
getting us?
Yes.
Thank you so much, sir. This was really an insightful presentation, highlighting the vitality and possibility of biotechnical tools and possibilities in disease management approaches. Your presentation was really delightful, uh, and your remarks on techniques like gene manipulation, recombinant technology, genetic engineering in disease management approaches has really broadened our knowledge on disease management strategies, be it on diagnosis, treatment, investigation of the abnormalities in genetic makeup, as well as in the detection of causative agents like bacteria, fungi, and viruses. Your highlightful remarks on the utility of polymerase chain reaction, which has been an inevitable tool in the detection of infection caused by SARS-CoV-2 that has confined most of us, as COVID-19 pandemics is also one of the glorious remarks that we have considered as one of the remarkable aspects of this conference. We are really grateful for your valuable inputs to our conference and expect that happens to be one of the remarkable part of this event. So we have questions which will be displayed on your screen. Please try to address the queries, sir.
Yes. If we have question, you can ask. You can read that question. If you think some questions are important, then you can ask. You can read that question. Your mic is muted.
Thank you, sir. Thank you for your excellent talk. It was really fruitful and informative. Sir, we have few questions with us.
Yes.
I request audience to ask the question. They can drop their question in the chat box. So there is one question by
Yes.
Chandrasekhar Devre, like how this recombinant technology can be useful to solve the problems of malnutrition and the locally available food.
No, no. I told that due to gene manipulation, scientists are producing high mineral containing food like golden rice, like iron containing. As you know, in there is anemic patient during pregnancy and during normal stage also. So many people are having a scarcity of that iron. So if we consume that high iron containing banana, so obviously we will not have that malnutrition due to iron deficiency. In golden rice, you have seen that vitamin A deficiency. So we have high amount of vitamin A in that golden rice. In this way, scientists are trying to produce the cereals or the food item which have high mineral content or high vitamins containing that food item, so that we don't have to take that pills of vitamin or pills of minerals. In this way, we can overcome that malnutrition.
Yes, thank you. Thank you so much, sir. So we'll have the next question, which
Yes.
has been asked by the Maninder. This is, if only we could include zoonotic viruses in our preventive plan.
Can you repeat what?
Sir, he is asking if only we could include zoonotic viruses in our preventive plan.
Preventive plan means as a vaccine.
Yeah, I
Preventive.
think. Yeah, I think he's asking for the vaccine as a preventive measure.
Now, as a prevention, we can use either complete virus or their subunit. Seed vaccine is a protein coded by that pathogen or virus. So we can take that gene which codes for surface protein, and we can use that protein as a vaccine. This is type of subunit vaccine in which we use recombinant surface coated protein of that pathogen, either vaccine, bacteria or virus.
Right. Yes, sir.
Yes.
So the next question is by Ujjwal La.
Yes.
Can you produce polyclonal antibody by using recombinant DNA technology? And what will be the source of this?
Yes, we can use, scientists are using, if we know, because polyclonal antibody means generally it's easy to produce polyclonal antibody. If we know monoclonal antibody gene sequence coding for that antibody, then we can clone that gene and produce that protein. Antibody is also protein, so we can produce in bacteria also, provided there are some glycosylations are not occurring in the bacteria, so we have to glycosylate. Without glycosylation also, efficacy of that antibody is similar to that glycosylated. So scientists are trying to produce monoclonal antibody. Polyclonal antibody means there are mixtures of antibodies. Then we have to clone all different. But monoclonal antibody through genetic engineering provided through fast display technology, we find out the exact gene sequence of that monoclonal antibody coding gene. Then we can heavy chain and light chain, we can clone their respective gene and then produce the recombinant protein in E. coli. Scientists are trying. I have also tried during my postdoc against that anthrax disease vaccine. Any other
Thank you.
question?
Yes, yes, sir. So there is next question by Satish Devkar. Please explain phage therapy and its potential for treatment.
Fast therapy means what? So attenuated, you are talking about attenuated phage virus. Fast therapy means what? I'm
So the
not,
question,
can he elaborate? Satish Devkarji, can you elaborate? Fast therapy means what?
please elaborate. Which kind
Phage
of
as a
phage
therapy.
therapy? Yes, sir. Like bacteriophages can be used as a therapeutic device.
Don't know, miss. How can we use phage? Phage we cannot introduce in human. Bacteriophage cannot survive in human. For human virus we use for therapy. For developing, we add that gene in that, suppose adeno associated virus and other virus. But bacteriophage we cannot use. Bacteriophage we cannot introduce in human.
Sir, I think the only gene of interest can be a clone in the phages, and then
Yes,
it
and
can
then we
be
can produce.
transduced in the bacterial cell culture, maybe
Yes,
for the better production of the
yes.
product of our interest.
For as a expression vector we can use as a phage sometimes. Generally for
Right,
library production we use phage, bacteriophage.
right, right. Yes, sir.
Am I, is
Yes,
it
we'll have.
convincible?
Yes,
Yes.
sir, we'll have the next question. Can you just tell me the cost of gene therapy, because it is quite costly? For that point, what is your opinion?
Okay, so depending upon the types of disease, some diseases are localized. Suppose blood-related disease, then ex vivo we can take that blood cell and transfect our desired gene, and then we can add. Suppose muscles-related disease, it's very difficult to change, manipulate complete entire differentiated muscle cells. Then it's difficult. So it's depending upon which disease you are targeting. Suppose some proteins are produced in blood, then we can target it easily. We can take out that cell, blood cell, and add our gene, and again we can add in, transplant into bone marrow cell that it can produce. But differentiated tissue, it's difficult.
Yes, sir. Will
And
just
exact costing, I cannot say exact costing.
yes, sir. So just quickly, we will have two more questions.
Yes.
This is why gene therapy is not so popular in treating the diseases.
What till gene therapy is not a popular method is treating disease? Why? Okay. So I told you, there are so many limitations. Something is ethical issue. Sometimes it's difficult. Not all genetic disease can be cured by gene therapy, because we cannot manipulate entire organism, entire human. If there is muscles related disease, then we cannot manipulate all the cells of the muscles in well grown human. It's difficult. Sometimes we are not getting volunteers also. Sometimes there are ethical issue also. That's why. But scientists are trying. Some countries have treated some disease using that gene therapy technology.
Yes, sir. Thank you. So
It's
we'll have
difficult.
probably the next question. This is the
Okay.
last question. Sir, how can we detect knockout gene by conventional PCR, especially for the heterozygous knockout gene?
Oh, very good. So if we can design primer against the flanking gene, so by suppose you have knocked out some gene X having 500 base pair, and we add some other gene. During replacement, we add some antibiotic resistance or some genes so that we can detect that cell. In that case, we can design a primer so that there is difference in size of amplicon with the non-knockout condition and with the knockout condition. So by seeing that, we can differentiate. And if you are talking about heterozygous condition, then by manipulation or by checking real time, you can check how many copies or there is any folded difference or not. If it is homologous knockout, both genes have been replaced, then concentration of amplicon after PCR will be different than the one gene replaced. So concentration will be different. In this way, we can distinguish. Otherwise, base method, sometimes we use that southern blot also, but PCR we can distinguish if we take proper control. Null mutant, heterozygous mutant, as well as non mutant, then we can see difference easily through PCR, or we can perform that real time PCR. So that number of copies we can differentiate. That is purpose of real time PCR. Am I right?
Yes, sir, exactly. So thank you so much. I think as we are running short of time and we have next speaker
So
in the
that's why I have rushed through, so that just I can give only insight.
yes, sir,
If
but you have beautifully explained and given the outline, like how the this recombinant DNA technology can be
yes,
useful for the
people should
development of
realize the potential of recombinant DNA technology.
yes,
That is
yes, sir, truly. Exactly. The motto has been served, I think. And thank you so much, sir, for your such a wonderful presentation and
thank you
being so
people also
attentive and
for
kind enough.
organizing this. Yes,
Thank you. Yes, sir. But we really appreciate your efforts and
yes.
just being so attentive and kind enough to answer all the queries raised by the audience. Thank you so much, sir.
Thank
Thank you
you,
for
ma'am.
being with us. Thank you.
Thank you.
Bye bye.
Should I leave?
Yes,
Should
sir.
I
Yes.
leave?
Yes, sir, yes.
Okay, thank you.
Yes. Hello, everyone. Now this is time to introduce our next speaker of the today's session. I request Samishka to continue with the introduction of our next speaker.
Thank you so much, madam. And we are again also thankful to our initial speaker. Dear all, after this intuitive lecture, we'd like to introduce with you our another invited speaker and one of my respected mentors, Dr. Tista Prasad Joshi. Let me introduce you to her. She is a scientist in Nepal Academy of Science and Technology NAST. She is consistently working in NAST since 16 years with a key focus on the drinking water quality research. She received an MSc degree in environmental microbiology and MA in anthropology in the year 2001 and 5 respectively from Tribhuvan University, Nepal, and holds a PhD degree in environmental engineering from University of China's Academy of Sciences, UCAS, China. Her research work associated with water quality and treatment has appeared in many leading journals such as Water Research, Chemical Engineering Journal, Journal of Hazardous Materials, Science of Total Environment, Chemosphere, RSC Advances, Frontiers of Environmental Science and Engineering. She has registered a patent in China and also writes several science awareness articles. She has presented her work in many national and international conferences. She mentors research works of postgraduate students and delivers guest lectures at university colleges in her field of expertise. Besides her academia and scientific exploration, she is also active in community level, governmental, and industrial consulting activities nationally. She has received a number of excellent and recognized international awards, including UCAS Excellent International Student Award 2017, OWSD LGV Foundation Award in Biological Sciences from Asia Pacific Region 2019, and OWSD Early Career Fellowship Award 2019. Let us welcome Madam Tista Prasad Joshi to deliver her lecture presentation entitled Wastewater Based Epidemiology Approach for COVID-19 Pandemics. Hello, Madam. Namaskar.
Namaste, good morning. Thank you for the nice introduction about me, Samiksha.
Thank you, ma'am. How are you, madam?
I'm good, and how about you?
Yeah, I'm also fine. Please proceed to the presentation.
Okay, and the slide, I'm waiting for the slide from the studio. Uh, okay, let me begin. However, I believe I'm a student of science. That is all my introduction. Thank you, organizer Sandip University and Biotechnology Society of Nepal for inviting me in this virtual conference. This is a good example how our scientific activities are adapting in with this global crisis.
Ma'am, I am sharing your PPT. Once I
Yeah,
share
yeah,
your
I'm waiting,
PPT,
and in the meanwhile, I'm also talking. I'll be
yes,
talking on
ma'am.
a different approach of understanding this pandemic. Before I start, let me clear you all valuable listeners. This talk is generally based on the literature review. I'm not presenting any of my research data here. At least, viewing the title of the presentation, you have correctly understood that I am trying to explain the relationship between wastewater and highly contagious COVID-19 disease, which is ongoing global pandemic. Next slide, please. Wastewater, in particular sewage, may reveal your secret of life. For example, what you eat, what you drink, what you are exposed to it, isn't it? Because the chemical in the form of the medicine, drugs, personal care product like cosmetic, alcohol, tobacco, different kinds of food are excreted as a whole or partially metabolized or completely metabolized form. And major concentration of these chemicals are shaded in sewage. However, the stability of the metabolites or chemicals in wastewater are dependent on various environmental factors such as temperatures, pH, and so on. Isn't it wonderful that you may know that how the community people are eating or drinking without asking them personally? So take a bottle of sewage and rush to the lab, know what's going on in your community. But hold on, you cannot pinpoint to a particular individual in a community for his or her secrecy. Instead, you can tell about a community, so this is a population-wide surveillance. In the same way, you think about the microbial pathogens. If they are colonized on body surfaces or cavities, including gastrointestinal tract, they will also be seeded in sewage. However, pathogenic microorganisms do not survive in a wastewater for a long time, or even if they would survive, their concentration would be below the detection limit because of continuous dilution. So, dear microbiologists, scientists, researchers, grab the sewage, detect the target pathogens, and know whether there is a current infection in the community or not. This approach of population-wide surveillance of public health issues is called the wastewater-based epidemiology. This concept is actually not a new, but known since few last decade, because this is very important by monitoring tool to explore the human exposure to the pollutants. Next slide, please. The idea was first proposed in the early 2000s when scientists began to explore it seriously, to explore the approach of tracking the used chemicals by the community or analyzing its wastewater. As you can see displayed on your screen, it was first proposed by Kristen Z. Dofton of the USA in 2001 in a book which was published by American Chemical Society. It was for tracking drug abuse in the society. Later it was published in 2005. It was applied in 2005. The concept was applied to monitor the cocaine, a very famous illicit drug in Italy by Ducato et al. In Italian, Ducato et al. In Italian cities. They analyze the wastewater and river water to estimate the cocaine concentration or cocaine consumption from the society, from the community. By 2011, many European countries applied and detect this approach to monitor the prevalence of illicit drugs around Europe. Again, by 2014, the same approach was successfully applied to survey the viral infections such as norovirus, poliovirus, hepatitis A virus, and adenovirus. WHO has also released a guideline for employing environmental sampling to monitor the polio in wastewater sample. And next slide, please. This literature provides a clear scientific evidence that emerging infectious viral disease can be monitored in the wastewater for public health surveillance or epidemiological study. As you can see in this graph, there is a clear peak of viral concentration in wastewater and the number of the patients diagnosed with the viral infection in the community of the sewage catchment area. The red line shows that the number of viral load in the sewage, and the black line indicated the number of the patients. As you can see, the peak of the viral concentration is followed by the peak of the number of the patients diagnosed with the norovirus. This means viral load. In fact, viral genome copy number indicates subsequent peak of the viral infection. So this relation can be applied to predict early warning of disease peak in the community. Many literatures, including this, have clearly indicated that some approach can also be applied in case of current global pandemic of COVID-19. And next slide, please. Now let us talk about a little bit about the coronavirus. COVID-19 is truly a global crisis. Every country, scientist, world institution, and in fact, everyone want its timely containment globally. Vaccine and the antiviral drugs are the best choices to no doubt. However, you are listening the warnings from the scientists and authorities that it will take time. So till then, we have to be protected. For protective measures and control should be followed, and there is no alternate. For protection and control, we need to stop spread of the virus, for which we must know who is infected and potential carrier that can be transmitted the virus to others. And how would we know who is infected? For this, we have to test. For whom you are going to test and how much your testing capacity, as you can see in this slide. The countries in our region are increasing their testing capacities. However, it is not enough. For example, in this slide please. For example, in this slide, you can see we are testing 2 to 3 individuals per 1,000 population. So, who are being tested? Those with travel history, or those who are contracted to infected ones, or those who have symptoms. The major problem of this disease is majority of the cases are asymptomatic. These asymptomatic cases possibly are not being traced and hidden in the community. However, they are spreading disease among their loved ones and others. In order to trace out this carrier population-wide surveillance based on wastewater or sewage monitoring is ideal choice. This is also important because of two reasons. Because testing capacity is too low and individual testing cost is also very high. And next slide, please. Hello, next slide. Well, so why do we believe wastewater-based epidemiology approach is very useful for population-wide surveillance of COVID-19? So two things are important. So does coronavirus can reach to the sewage and aphids, how long it survived there? This Chinese study published in one of the Lancet publication journal on March of this year clearly shows that the virus is also shedded in faces of the majority of the infected individuals. Here in this graph, this yellow bar, hope you can see in the slide, the yellow bar represents the presence of SARS-CoV viral in the fecal sample of the hospitalized patient shown in this graph. This is the true evidence that the coronavirus can appear in sewage via fecal matter. And next slide, please. In fact, RNA of SARS coronavirus 2 is present, has been detected in wastewater of many countries such as the Netherlands, the USA, France, Australia, and Sweden. Sewage sample of 7 cities and the airport were analyzing using the RT-PCR, and SARS coronavirus 2 was detected in the sewage. This is also proof that sewage surveillance could be a sensitive tool to monitor the transmission of the virus in the population. Next slide, please. Similarly, report from the Australia, which has been published in the Science of the Total Environment Journal, it clearly showed that the presence of SARS coronavirus 2 in the wastewater. This was the first reported case detection of the SARS-CoV-2 virus in wastewater in Australia. The presence of SARS coronavirus 2 was confirmed by sequencing. So wastewater-based epidemiology is an innovative approach to understand the prevalence of viruses, prevalence of diseases in the community for the population-wide surveillance of COVID-19. Authors concluded that the wastewater contains virus excreted from asymptomatic individual in a catchment, symptomatic and asymptomatic individual in a catchment. Please note that Australia is using a wastewater-based epidemiology approach for the surveillance of illicit drug abuse in the society. So now they are approaching same strategy to solve the coronavirus infection, analyzing a wastewater. They believe this approach is crucial for COVID-19 containment in Australia. Next slide, please. Here is another story of the Paris. By sampling sewage across the Greater Paris for more than 1 month, researchers have detected a rise and fall in novel coronavirus concentration that correspond to the shape of the COVID-19. Higher concentration of virus in the wastewater correspond to the higher number of infected people who contribute to the sewage system. You may remember this research emerged when there was a peak of COVID-19 cases in France. Once they found SARS coronavirus 2 RNA in wastewater. They sealed whole catchment community and extensively tested the individual in the community, which helped greatly to control the coronavirus. Probably this declined the curve in France. Now I'm talking about the second question is how long this virus sustain in the sewage. Next slide, please. The primary mode of transmission is via respiratory droplet or bioformites. There are different reports regarding the lifespan of coronavirus in the environment. Many reports agree that the coronavirus can survive in sewage for at least 3 days, whereas for the solid feces, it can be survived for more than 3 or 4 days. Next slide. However, different environmental factors may influence the survivability of the virus in sewage. For example, disinfectant including sodium hypochlorite and other oxidants can lower the stability of the virus in the environment. It is also known that coronavirus appears to be inactivated significantly faster in water than non-enveloped human enteric viruses. And temperature is an important factor influencing a viral survival in the environment like sewage. It is reported that the titer of the virus declined more rapidly at 23 to 25 degrees centigrade than at 4 degrees centigrade. Commonly, sewage has approximately same temperature, 23 to 25 degrees centigrade, though it may depend upon the seasonal and geographic variation. Hence, coronavirus is generally considered unstable in the environment and is more susceptible to oxidants such as chlorine than non-enveloped viruses like the polioviruses, enteric viruses. Similarly, it is activated by the ozonation process, multiple barrier reactors, chlorination, advanced oxidation process, ultraviolet inactivation process, and so on. Here it is good news that the disinfection process during the wastewater treatment can eliminate or reduce the coronavirus from the wastewater. Broadly, you can say from the environment. And next slide, please. Here is another example that here is general method of wastewater-based epidemiological survey of the infectious disease, including virus. It can also be used for the population-wide surveillance of COVID-19. In fact, both are same thing. The wastewater coming out of the community, for example, apartment, can be sampled either at sewerage system or more conveniently at wastewater treatment plant. Because mostly sewerage pipeline may be closed type, so sample could be better sample at treatment plant. Collected water sample can be transported to the laboratory by viral detection and quantitation by using appropriate molecular technique. For example, real-time quantitative PCR. However, molecular method may depend upon your laboratory capacities and sewage factors because complexity of wastewater matrix is sometimes challenging for extraction and quantification of nucleic acid as sewage may contain various PCR inhibitors which may be coexisted with a viral nucleic acid. Some advanced molecular biology techniques offer a new route of genetic material analysis, for example, digital PCR and next generation sequencing with metagenomic sequencing and strategies. If you detect the viral RNA in the sewage, indicate that the individual in the sewage catchment community may be infected. This information can be transferred to public health officials or concerned authorities who will test the individual in the community. Once tested, positive individual can be isolated or admitted to a hospital. So, they could not be transmitted the virus to others, and of course, infection be managed by treated infected one. Actually, this is the whole process in brief. Next slide, please. However, this is a multidisciplinary approach where a number of resources with different expertise work together. So you can see the scientists are turning now to wastewater-based epidemiology as a potential tool for assessing and managing the pandemic. These data are vital for health impact assessment and to support the environmental and health policy making in public health program. Therefore, monitoring wastewater has the potential to extract the useful epidemiological information from qualitative and quantitative profiling of biomarkers entering the sewage system. Next slide, please. Actually, scientists have started publishing their work on wastewater-based epidemiology of COVID-19. For instance, let me show you one example. Jordan Peckay and colleague from Yale University of the United States of America has recently published a paper. In this paper, they reported a time course of SARS coronavirus 2 RNA concentration in primary sewage loss during the spring COVID-19 outbreak in northeastern American state of the metropolitan city area. SARS coronavirus was detected in all environmental samples. And as you can see in the given graph, when adjusted for the time lag, the virus concentration are highly correlated with the COVID-19 epidemiological curve and local hospital admission. The figure A here shows that average loss SARS coronavirus RNA concentration time course data, which represent the red color, and average hospital admission data represent the black color in this peak, with the peak at the same, both at the same time course. Another example of the figure now, graph B, is scaled at the smooth SARS coronavirus RNA concentration in red, and hospitalized patient admission patient is in black color. Here in the graph number C, smooth SARS-CoV RNA concentration with red color with smoothed COVID-19 epidemiology curve is in black color. As you can see, figure D and E, the data can be used for the regression analysis that can predict the proportional rise in number of the new infections. And next slide, please. Okay. Wastewater-based epidemiology in Nepalese context. Actually, this context is very new context in case of the Nepal. Wastewater-based epidemiology is not implemented so far in Nepal, and most of the literature reveals of the pathogens so that there is a enteric pathogen, antibiotic-resistant bacteria, human viruses, but feasible and must be applied for population-based surveillance. And this municipal wastewater treatment facilities are also limited in Nepal because there is very less than 10% of total generated wastewater are used for the treatment. However, many treatment plants are under constructed in the provincial level. And here is some possible wastewater based epidemiology sites are sewage wastewater treatment plant, hospital wastewater generating plant, likewise hospital health care treating COVID-19 patient, and also the quarantine isolation site. Next slide, please. Okay, it is important for water engineers and professionals to understand the nature and fate of coronavirus and effective measures to protect the public health. There is no evidence to suggest that coronavirus are present in surface or groundwater sources or transmitted through contaminated drinking water and also from the food. Further researches are needed to study the potential presence and fate of coronavirus and other enveloped viruses in municipal wastewater and drinking water to provide evidence necessary for the prevention of future outbreaks. More research work is required to investigate the potential for the viability and effectivity of SARS coronavirus 2 infections. And next slide, please. These are some references because this presentation is generally based on the literature review, and none of the data are not mine. And I would like to acknowledge all the researchers, contributors, scientists for this great contribution for the society. Because of their valuable research, I'm here to present you in front of you and share some of the this important view on this topic. Uh, and next slide. And thank you. I would like to thank my academia and thank you, this organizing committee, for inviting me. And thank you all for joining me here. Thank you so much.
Thank you so much, madam, for your delightful reflection on several aspects of water quality researches and its relatedness to various possibilities of disease study. Since water is the fundamental requirement of living system, contributors like you who are focused on the monitoring of water quality issues and seems to be inevitable part to bring about improvement in the aspects as remediation approach in water quality. Since the virus infecting human have already been identified in water resources and sewages from different nations around the world, I hope the connection that you mentioned about the necessity of water quality testing and sewage management during this pandemic time stands out to be one of the most impactful remarks that this conference will be drawing. Along with this, your crucial views on correlated management of infrastructure in society with a positive message in every aspect being talked are really motivating and also seems to be an issue of immense consideration by authorities and to those researchers wishing to work in wastewater quality as well as drinking water quality. With this note, we express our gratitude towards your valuable insight expressed enthusiastically to the audience watching us. Now, I'd like to hand over to Dr. Raitekar Madam to take over some questions from the audience.
Thank you, Samiksha. Thank you very much.
Thank you so much, ma'am. Thank you
Thank you,
very much
Dr.
for
Namita.
your, yeah, thank you, ma'am, for this wonderful lecture. We will quickly have few questions.
Yes.
There is a question by Sujata Dungel. How country like Nepal can proceed sewage surveillance for COVID-19? Any suggestion, ma'am?
Yeah, as I mentioned during my presentation, also the context of the Nepal, it is feasible and should be done by us. It is not so difficult actually, though currently also, but the labs are closed because of the lab. Currently, we cannot do because of the labs are closed because of the lockdown. But it is feasible, and if all the equipment is available and all the process is available, I think it is we can do in the laboratory also.
Yes, thank you, ma'am.
And
The next question is there any possibility that COVID-19 can be transmitted through drinking water?
as I have already talked about this also, till now there is no research that there is no any evidence that the COVID-19 can be directly transmitted to the drinking water. This is also another aspect of the research, that whether it is transmitted or not, because this virus is totally new for us. And the researchers are everywhere are engaged to do the research on it. And also there is no any kinds of relationships of the transmission of the from the food also till now.
Yes, ma'am. Thank you so much. Ma'am, there is one more question. Like,
Yeah.
how these molecular methods are reliable? Like, if we just, because they are based on the gene detection, and if we just detect them, if we are able to detect any gene in the environmental sample, and the pathogen is actually so, like, how reliable this method?
I didn't hear you.
Hello.
Yeah. It's not clear. Could you please?
Hello, can you hear me?
Yeah.
Hello, ma'am.
Now I'm hearing you. Yeah, Dr.
Yeah.
Nomita.
Yes,
Could you
ma'am.
please
The
repeat the
question,
question?
yeah, I will repeat it for you. Ma'am, the question is like how these molecular methods are reliable if we are looking for the human infectious agent and their traces, the genetic traces have been detected in the environmental sample, then how it is correlated?
Actually, you know that the complexity of wastewater matrix is sometimes very complex and also challenging for extraction and quantification of the nucleic acid, as sewage may contain the various PCR inhibitors or many interferences also. And it also depends upon the some kinds of the kits, reagent, some and others aspect. But it is, however, it is reliable, and most of this molecular method have a good result, in my opinion.
Yeah. And ma'am, do you think that there is any chance that the this genetic material from the human infectious agent can be transmitted into the airborne infectious agent? Is it possible?
Airborne, I'm not sure about whether it is possible or not to make, because for that also we have to look on the literatures and the research. I'm not familiar with this. It may be or may not be. I'm not sure.
Yeah, no problem. Thank you so much, ma'am. Uh, I request audience if there are any questions, they can immediately drop it so that we can consider those questions. I think, ma'am, there are no more questions from the audience
Okay.
side. And thank you so much for being with us today and
Thank you,
being
Dr. Namita.
attentive. Yeah, and answering all the questions with a lot of patience. Thank you so much, ma'am, for your informative talk. Nice to see
Okay.
you.
Yeah. Namaste. Thank
Yeah.
you.
Good day, ma'am.
Okay.
Bye bye.
Bye.
Yes. I think with this we can conclude that today's session is over and we can go with the presentation session. The first round of all the speakers that has been concluded, and with this we can start with the next session of the presentation. I request all the presenter to be ready with their presentations. We will have 5 minutes break and immediately after that we will continue with our presentation. Thank you.